Okay. It's going?
It's going.
Okay, great. Good morning. My name is Fred Heinzel. I'm from the Department of Internal Medicine at USF Health, and I would like to talk about T cells, specifically focusing on the many different types of phenotypes and the immunologic functions of T cells. Well, first off, starting at the simple part first, comparing B cells to T cells. T cells recognize T cell antigens that are almost nearly all proteins and are derived from the thymic tissue. The receptors are alpha beta T cell receptors on the T cell surface, which recognize peptides incorporated into MHC2 and MHC1 histocompatibility. Okay. I need to see the transitions. Okay. All right. So they recognize alpha beta T cell receptors on T cell surface. They recognize peptides incorporated into MHC2 and MHC1. That's for T cells. Now B cells recognize proteins, nucleic acids, polysaccharides, and other chemicals, which is in contradistinction to T cells, which mostly see only peptides in MHC compatibility antigens. Okay, the origins of T cells arise from the thymus. The thymic cells are normally double positive when they're first developed. When they bind to MHC1 or MHC2, they become single positive for CD4 and CD8. So that's really where the CD4 and CD8 cells arise from. So mature T cell subsets are typically CD4 positive T helper cells. Again, they have alpha beta T cell receptors that recognize antigen in the context of peptide in MHC2. The CD4 molecule actually augments activation. Now, the CD4 molecule is provided by the CD4 cell as it nestles up into these complexes, and the CD4 molecule actually activates and stabilizes the MHC2 peptide TCR complex. CD8 cells do a similar thing in that the CD8 antigen on the surface of the T cell also augments and stabilizes activation of the MHC1 peptide TCR complex. So that's why MHC1 and MHC2 seem to segment into either CD8 cell or CD4 cell interactions. Now there's another so for MHC1 and MHC2 processing, remember MHC1 processing is for CD8 cells, and MHC2 processing is for CD4 cells. The pathways for display of antigens are processed from within the cell or sometimes outside the cell. So on the left-hand side, we're looking at one situation which is unique only for CD8 cells and MHC1 class 1, what you see on the screen here, which all the MHC class 1s all have a beta 2 microglobulin that kind of tags the MHC1 class down to the cell surface. What the CD8 T cell processing system does is it samples the endogenous antigens in the cell. And these can include viruses, which are in the cytoplasm, and these proteins, large proteins, actually get chewed up by a proteasome, which you see in the middle there. And those antigenic peptides go in through these transporters into the endoplasmic reticulum. So the antigens that are going to be processed by MHC1 to CD8 are all based from peptides generated within the endoplasmic reticulum. In contrast, processing of antigens in the MHC2 processing pathway is considered an exogenous pathway for display of antigens. So in this case, on the right-hand side, MHC class 2 is expressed on the cell surface, and uptake of exogenous antigen it goes through phagal lysosomes, endosomes, and goes into a variety of vacuoles that transport the peptides onto the HLA-DM and HLA-DO system. There's also a little peptide called CLIP, which actually blocks the uptake of antigen, which is probably a mechanism that assures that if to basically preserve open spaces for binding peptides. Right. So in this case, this is exogenous because all these peptides are all coming from outside the cell. So this is going back again. For the class 1 system, you're only looking at peptides generated within the cell cytoplasm. Whereas in the class 2 processing, all of these antigens and peptides are being generated through uptake of the external surface and processing and placement of these. Okay, and placing these peptides into HLA molecules. Okay, so when the T cell receptor meets up with the MHC2, so what you see on the left-hand side on the top, you have MHC2 class, which is actually on both top sides. On the left-hand side, you can see where you have alpha 2 beta 2, alpha 1 beta 1. You have the little red antigen right there. So that's basically where the antigen fits into the MHC1 class. And down in the bottom there, you can see those sort of complementary little chunks there, and that's the T cell receptor. So this defines the MHC1 class 2 binding of the antigen, which in turn is incorporated into T cell receptor that allows the T cell to activate and make cytokines. And on the left hand, we're looking at antigen alone, but there's also something called superantigens, and this is in the situation of a toxic shock syndrome, where you have proteins that are capable of not binding in the complementary region between the MHC2 and the T cell receptor. Instead, what's happening, it seems to stick to the outside, and so it binds T cell receptor and MHC2 class in sort of an artificial fashion that results in a form of activation that's very potent, and these patients will have increased levels of TNF, interleukin 1, and so I mean this is classic for staphylococcal shock syndrome. Okay, so that's really two completely different ways of activating the cell. Typically, it's the T cell receptor that does that. Superantigen is just sort of an odd variant. Okay, so on the top, you can see where the green peptide fits into the into the MHC molecule, and there's a groove in the MHC molecule that the peptide can fit into. In the case of MHC1, the peptides are cleaved at about 6 to 10 amino acid residues, so they're relatively short. And if you look at this right here, there's some hindrance on either end of this open cleft here, and which ensures that they pick up these small peptides that are being cleaved by the endogenous system in the cell. Once the T cell activates with an accessory cell or some other cell, there is an immunologic synapse that occurs during T cell receptor antigen activation in conjunction with MHC interactions. And as you can see here, on the bottom you've got the antigen presenting cell, On the top, you've got the T lymphocyte, and you can see all these complementary regions here. Each of these have specific functions and are very important in establishing a phenotype for the T cell. They increase the affinity of activation, and you can also get some diversity of cytokine production or T cell phenotype differentiation. So, and most of these are the complementary parts, of course, would be like CD2 to LFA3, and there's a variety of other molecules that go along with that. So again, it's like a synapse. Everything gets together. There's like a zipper-like effect that activates the T cell and provides information as to how it's going to differentiate over time. There are multiple CD4 Th1 Th subsets, and they all exhibit distinct cytokine profiles. For instance, Th1 produces interleukin-2, produces lymphotoxin and interferon gamma producing cells. Interleukin-2 is generally a growth factor for T cells, and they can drive proliferation of any number of CD4 and CD8 T cells. The Th2 phenotype produces the cytokines of interleukin-4, interleukin-3, and interleukin-5. Interleukin-4 is kind of the classic allergy-inducing cytokine, and interleukin-5 is actually a cell growth factor for eosinophils. So the Th2 program there is really something that is a part of it. It's an allergic program. More recently, the Th17 subset has been described, and I'm going to talk a little more about that a bit later. And then I'm also going to talk about T regulatory cells as well, which is both the Th17 and T regulatory cells are really sort of the new kids on the block, but are very important. So. So what some of the common themes in T cell differentiation is activation of certain programs by tyrosine kinases and phosphatases and all of these little sort of miniature chemical machines that are working in the T cell. In this case, we have two kinases. One is STAT6, the other is STAT4. STAT4 drives a Th1 T cell program. STAT6 drives a Th2 program. So there's this sort of a divergence of T cell phenotype based on the kinases that they have. Okay, this is I'll try and abbreviate this a little bit. So we talked about the Th2 cells. We talked about we recognize that there are environmental factors and genetic factors that also act in concert to determine the Th1 and Th2 polarization. So getting down to pathology here, Th1-dominated responses are involved in the pathogenesis of organ-specific autoimmune diseases, Crohn's disease, sarcoidosis, kidney allograft rejection, and unexplained recurrent abortions, in part because the Th1 cytokine activates interferon gamma and possibly some lymphotoxic cells, and so this can cause deleterious inflammation. In contrast, allergen-specific Th2 responses are responsible for atopic disorders in genetically susceptible individuals. Th2-dominated responses play a pathogenic role in more progressive systemic sclerosis, which we now know that interleukin-6 is actually probably the causative cytokine that causes systemic sclerosis, and their efforts now is trying to neutralize interleukin-6 in scleroderma. Okay. And finally, the Th1 and Th2 paradigm can provide the basis for the development of new types of vaccines against infectious agents and of novel strategies for the therapy of allergic and autoimmune diseases. So there's a lot going on there. And just even the Th1 and Th2 hypothesis has had kind of a long lifestyle and is still pretty active. Okay, so other examples of CD4 Th1 function is that dendritic cells, when they see activated by toll receptors or activated by other stimuli produce interleukin 12. Interleukin 12 induces interferon gamma synthesis by Th1 cells that activates nitric oxide synthase. That is essential for killing the intracellular and extracellular pathogens. Recognize that nitric oxide synthase produces nitric oxide, a diffusible gas, goes through the outer membrane. It can get into the internal endosomes and lysosomes, and it is highly toxic to parasites, bacteria, and a variety of other pathogens. So that's actually a very important defense strategy for the host. So we need Th2 cells and Th1 cells and interleukin 12 for that. Okay. So CD4 Th1 function consists of activation of subepithelial mast cells in gut and respiratory tract, production of IgE that promotes asthma and other allergic reactions, including eosinophilia, goblet cell hyperplasia, and immediate allergic responses. In short, Th2 activity tends to drive asthma, possibly eczema to some extent, and other allergic reactions. The role of eosinophilia in control of helminthic infections has been disputed for a long time. There were some older studies suggesting that eosinophils were able to kill helminths, but that's not as well supported as it used to, just in case you came across that at some point. Okay, since 2007, there's a new player in the field. This is the Th17 cell, so named because it makes the cytokine interleukin 17. It starts as a native naive CD4 T cell under an exposure of a variety of cytokines such as TGF-beta, interleukin beta, and then there's a all of these T cells now they have signaling cascades that include STATs, which are kinases, and the RRO gamma is actually another transcriptional factor. And these then define the T cell phenotype. And okay, so once you get the Th17 cell, it begins to divide. The terminal differentiation and expansion stage. So what does the Th17 cell do? The release of interleukin 17 induces chemokine receptor expression on various tissues and recruits T cells into sites of inflammation. Th17 cells specifically contribute to tissue inflammation and injury. Th17 associated pathologies include juvenile diabetes, rheumatoid arthritis, multiple sclerosis, sarcoidosis, and Crohn's disease. As you could expect, there's a lot of interest in making humanized antibodies against Th17 and against interleukin 23, which is also contributory to Th17. And some of those I think are in market already. So, but the interesting question here is, how does CD4 Th cell diversity develop? So you start out with single cells, and at some point they develop this huge range of activities. And just to give you sort of an idea of this, is you get a naive CD4 cell, you activate it, and under certain circumstances you get a Th1, a Th2, Th9, we won't talk about that, Th17, we did talk about that. There's a 22, there's a T regulatory cell, and the TFH, actually I don't recall what that is, that's something new. So it makes interleukin 21, I guess, so that actually would be a B cell factor. So you got all these cells to make a lot of cytokines, and the sort of things they do, you enhance the survival of eosinophils and progenitors. You get IgE, B cell immunity, and so. But the interesting thing here is all these cells come basically from supposedly a single primordial T cell, and how do you get this incredible range of activities? One idea that is current is that this all depends on the presence of cytokine receptor associated kinases within the cell, which some people call the tiny machines of signal transduction. And as T cells divide, as T cells divide, you will actually segregate Janus-associated kinases, which is a kinase that's very important in differentiating cells, and the STATs, which is also important in differentiated cells. And the cytoplasm may segregate those factors into the daughter cells, and that provides almost sort of a random mixing of all these factors, and it's suggested that that's an important part of this. And actually, the model for this is actually a pachinko machine, which is pachinko machine, which is a Japanese gambling device. So what happens is you drop your balls down in here. You see these? You drop these little balls down in there, and every time they hit a peg, they make a decision. They either go to the left, they go to the right. And you keep on doing this, and as you go down, all these little balls sort of that down there. So now it's a model of just basically binary division of these cells. Reassorting all these things, you get a continuum of different phenotypes of this. Okay, but it isn't enough. You're at that point, you're just setting up the diversity. What happens after this is the dividing T cell precursors segregate tyrosine kinase and other, and it's all done in a probabilistic fashion, which just means random. So the T cells, if they're predisposed, if they actually express a receptor and are predisposed to receptor activation by cytokines, they will opportunistically expand into defined Th subsets, which is really a survival of the fittest, and this is really kind of apologies to Darwin, because this is pretty Darwinian. Okay, so this raises another question. I mean, so now we have all these T cell phenotypes, and we're just scratching the edge of this. There's probably 3 times more phenotypes than we know about, and we just haven't really identified all of them. So you have all these cells. Most of them are pro-inflammatory. They make all these cytokines. So how do we avoid just being a big raging ball of inflammation? That has always been a puzzle. There's a lot. For years, they were talking about suppressor cells, and this was really big at the University of Chicago. They were kind of like the world mecca for suppressor cells, and they got most of it right. But over a period of time, it's become apparent that it's a little more. The thought is just that some of these T cells were somehow would magically suppress, activated pro-inflammatory cells. Okay, but what we now know, and this was that we have T cells that inhibit other T cells, and these are called T regulatory cells. They attenuate pro-inflammatory responses, in part because they make all sorts of little goodies off to the side here. I'm not going to go through all of them, and I'm going to have another little slide that's going to kind of focus on that more. What it is the T regulatory cells very immunosuppressive. There are all these interactions over here. A lot of these actually involves blocking activation of T cells. And I'll go on to the next one here. Okay, so T cell regulatory cells. It's a subpopulation of T cells that modulate autoimmune disease and cancer, and then they can facilitate organ transplantation. The phenotype is CD4 positive. Okay, for T cell. There are also these T regulatory cells are also CD25 positive. Now that's the interleukin-2 receptor. For some reason, that probably contributes to an inhibitory phenotype. And you also have a transcriptional factor called FOXP3. I think we talked about that once before too. These T regulatory cells also mediate antigenic tolerance and suppression. You can identify the T regulatory cells if you basically take a mouse spleen, put it in, to tease apart all the T cells. You can use flow cytometry. I mean, some of you made flow cytometry. Rings a bell? Okay. So what happens here then is that for the T regulatory cell, you have CD4 that's marked with FISI, which is a green dye. And it's over this way. So that column right there are CD4 cells. But then CD25 is pointing up that way. So you know that in that group of CD4 cells, there's a significant subset of CD25 positive cells. And so that little upper quadrant is where your T regulatory cells are. And you can actually sort those things out. You can put them into dishes and play games with them. But that's relatively recent. It was probably over last 5 years that this really came to fruition. So it makes a lot of sense at this point. We always knew there had to be some way to turn off the inflammation. And by that, if we can understand the T regulatory cells, we may in fact be able to use them therapeutically in autoimmune diseases. And actually that's really kind of picking up these days. Okay, so there are a lot of immunomodulatory, anti-inflammatory cytokines. Tumor necrosis factor can actually cause some cells to die. That's one way of taking care of inflammation. Interleukin 10 is sort of a downer. It tends to turn off cytokines. I don't know about interleukin 12. I think interleukin 12 sometimes there's a feedback loop that can actually kill other cells. So you got a range of things here, and again interleukin 10 here, which is a highly immunosuppressive cytokine. So that supposedly tips the anti-inflammatory process the other direction. Okay. So how do we know T regulatory cells actually prevent autoimmunity? Genetic deficiency of FOXP3, which is an important factor in making a T-reg cell, results in overwhelming autoimmunity. It's called the IPEX syndrome, which is a syndrome for immunodysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome, which is pretty devastating. It's X-linked, so of course it's only males. Affected males develop psoriasis, alopecia, diabetes, and bullous pemphigoid. Now, there's therapeutic uses for expander T cell regulatory cells. Again, here we're back to this gate here, and what you do is you can gate out the CD25 cells, and you interact them with MHC2 class, peptide T cell receptors, and sometimes you can get actually antigen specific suppression through the T regulatory mechanism. So there's an increasing trend towards clinical use of T regulatory cells. In order to do this with humans, you got to bank PBMCs. You got to then use the donor spleen cells, and you incubate them with thymoglobulin and sirolimus, which is one of the immune suppressive drugs for graft rejection. And these actually show evidence of being able to suppress inflammation. So in this case, this is for diabetes. Okay, so on the right lower side here, T-regs, if you put them into a culture system with insulin secreting cells, you can actually reverse diabetes. So basically by downregulating the inflammation. Okay. So antigen-specific T-regs reverse diabetes by blocking pathogenic T-cell differentiation. Okay, so this is kind of the proof of principle. These murine data demonstrate the clinical relevance of T regulatory cells in autoimmunity and graft versus host disease. Now, this also feeds a little bit into the hygiene hypothesis, which most of us have talked about a couple times in the last couple years, and which is with certain signals, namely helminthic signals and bacterial signals, you developed an unbalanced expression of Th1 cells, which are proinflammatory, and Th2 cells, which are really allergy-inducing. The thought now is that you can modulate all of this if you can introduce T regulatory cells. And at this point, you don't get either immunopathology. The idea is that this sort of puts it on neutral ground. And which also is going to prevent autoimmunity. Okay, so just a few other things here that are just sort of odd that I picked up on. I thought they were very interesting. Is there's several cancer centers are now using immunotherapy using soluble antigen receptors. These are soluble B antigen receptors. These are being used to target antigens on cancer, and you can even use them sometimes for graft versus host disease, because you can basically jigger the VH and the VO any way you want, and then you can target tumor antigens on the cancers and make them go away, theoretically. Okay, and finally, there's also cancer immunotherapy and therapeutic vaccines. The Provenge, or the Sipula cell T, consists of autologous dendritic cells. Dendritic cells, of course, are cells that are highly potent in presenting MHC2 and antigens and cytokines. You obtain the cells by leukapheresis. You add a blood product that is incubated with a fusion protein, which binds, so that makes the cells all sort of join together. The antigen that they're targeting is prostatic acid phosphatase. Basically, this is a drug that's aimed at treating prostatic cell cancer. And you also provide GM-CSF cytokine, which activates APC in dendritic cells and increases the potency of the inflammatory product reaction. The blood product is reinfused into the patient, and the IMPACT study that was a major study showed significant benefit for the treatment of prostatic cell cancer. So just pointing out that cytokines. T cells, cytokines, and including B cells can all contribute. All right. So I think there's a whole new world out there. First off, a lot of these T cell subsets, we don't know much about them. We're going to probably identify some more. We can manipulate these TH subsets mostly by using humanized antibodies against distinct cytokines, and that's already been done. If you look at Stelara and Enbrel and all those things, I mean, there's a whole marketplace out there, and there's a lot of immunologists being very hyperkinetic and waving their hands about this. Leukin 17 already, there's a lot of efforts to try and tackle that, because again, it's probably one of the big enchiladas of organ inflammation. So it's not. This is mostly TH. This is mostly T cell related, but it's because the cytokines can be neutralized by antibodies. You can manipulate all of these, hopefully in a therapeutic fashion.